Uh, at 8 p.m. I will invite you as with professional intro. Okay.
ओके। दिस टाइम इस टाइम देखें, ये एक स्लाइड शो हो रही है या दो?
ये 2 स्लाइड हैं।
मतलब साइड रिव्यू व्यू भी शो हो रहा है?
जी जी।
ओके।
नाउ अब ठीक है।
ओके, ये अब ठीक है।
जी जी।
ओके, सही है, इसको क्लोज कर दें?
जी आप स्लाइड पे आ जाएं। स्लाइड।
हाँ जी। हेलो।
जी जी, मेरी आवाज़ आ रही है।
स्लाइड शो हो रही है?
जी। सेकंड स्लाइड, अब फर्स्ट स्लाइड शो हो रही है।
हाँ
सेकंड
जी।
स्लाइड। फर्स्ट स्लाइड पे चले जाएं। यस। नाउ
आपको कौन से
फर्स्ट।
स्लाइड दे चाहिए?
अभी फर्स्ट ही चलेंगे ना। हम फर्स्ट स्लाइड से
ओके।
जी। Brother, we will start within 2 minutes.
Okay, okay. Can you hear me?
Yes, your voice is audible.
Okay. Now my voice is clear. Hello, my voice is clear.
Okay. Assalamu alaikum everyone. I hope you all are doing well. So welcome in today's class, primer designing strategies for efficient gene construction. Here Gulam Murtaza and our today's guest speaker, Dr. Naeem. He is currently doctoral student at Tianjin University, China. And welcome, Dr. Naeem, on in
Thank you.
today's uh, internship session. Uh, Floor is for you. Please go ahead. Thank you so much.
Okay, thank you. Hello everyone. My name is Muhammad Naeem. I am a PhD student in Tianjin University of China. So today I will present my lecture on primer design strategies for efficient gene construction. First of all, we will discuss about the primer designing through a snap gene. Snap gene basically is a software and that allow to the user visualize and annotate the DNA sequencing. It can be used to plan the cloning experiment and PCR strategies by using the different kinds of PCR like real-time PCR, qPCR, and simple PCR. So for the primer designing, the basic needs we should have a DNA sequence first that is known as the gene, and the primer length also should be 18 to 20 nucleotide bases, and the melting temperature should be around 55 to 65 degrees centigrade, and GC content, that is the very important things in the primer designing, so it should be 40 to 60%. And final important thing is the hairpins and the dimers. We should must avoid the hairpins and dimers. So next part is the gene construction and the purpose of the gene construction. So it's mean why we are constructing the gene and what's the construction. Gene construction is the construct is a basically is the is contain the gene and vector. Here you can see there are different genes, and this whole is known as the vector. Uh, so. The main function of the construct to express the gene in the cell or organism, and the second is the to study the protein function and interactions of the genes, and the third one is the gene regulations. If you want to check the functions of the gene, then you should express a gene in a specific vector, and last one is the to perform the gene editing or RNA interference. If you want to edit or any genes and you want to change something in the gene, then you need to introduce your gene in a specific vector, and then you can express and check the expression of the relevant gene. So today I will tell you how you can design or construct. So for the gene construction, first of all you should must have a vector, and then you should have a gene of interest, and third one you must should have a snap gene setup. So today I will use the PET28A vector and PGM1 gene for this practical work. So in this diagram, this blue color is showing the vector, and this color is showing the this color is showing the fragment mean gene of interest. So when you want to introduce a specific gene in a vector, then you must should you must should digest the specific vector, and for the digestion you should use the two specific restriction enzyme which we will use in the practical session. And these you can see here these red color in vector and in gene of interest, these points are showing the gene are showing the restriction sites, because genes and vectors both will be integrated with these points. And finally here you can see a successful vector, and when you have a specific vector and then you can this is known as the construct. And finally then you can when you have a construct, then you can transform in the bacterial cells or another specific vector or genes. So now we are going to explain how we can do this. Can you hear my screen? Hello?
Yes, your screen is
Uh,
visible. Please continue.
okay. Okay, thank you. So, first of all, we need a gene and vector. So, I already have a vector, PET28 vector, and now we need a specific sequence of the gene. So, I am using the PGM1 gene, so I will use the NCBI to download the sequence. So, maybe NCBI is not working, so we can use the UniProt, and over gene is the PGM1. You can use any gene, and here we need a sequence, and here this is the protein sequence. This gene, when this gene will express, it will show the specific protein, and this one is showing the protein sequence. So we need a nucleotide sequence. So we will go the DDBJ and we will collect the specific sequence. So here it's showing the CDS sequence, and we will copy this sequence and open the snap gene, and click on file and open a new DNA or RNA file. If you have RNA file, you should click on RNA, but we have a DNA sequence, so we will open paste here, and we will give a name here PGM1, and okay. And now we have a sequence, and we will select the all sequence and go to the feature and add a feature. And here we can add a specific name PGM1, and here features must should be CDS, and here should be product. And here you can change the color of your fragments. And now here you can open the frame for the proteins, and here you can check your proteins sequence. This gene is expressing these proteins, so it must should be these proteins inside the gene. So control F, and here you can check the protein sequences. So over sequencing is starting from this M. Here we will use the just one frame. Okay, anyway, yeah. So our gene is starting from this M. So M is methionine, which is starting the any expression system. So these are all the extra nucleotides. So we should delete these nucleotides. And similarly, so at the end, if you have some extra amino acid, we also must should delete this from the KVKK. After this, we must should delete this sequence because this is extra sequence. So the starting region is M double AY and the end session is VKK. So we will confirm here. Here we can see the starting region is MAAY and last region is KVKK. So it's mean our gene is ready for primer design. So first we will click on this PGM1 mention name and we will copy this control C and now we will design the primer. Here we will go uh, Google and we will use here prime plus 3 prime 3 plus and this website you should open and paste your sequence your DNA sequence and here you should pick the colony primer and pick primer and now this is your forward primer and we'll copy this primer and here go to the primers and add primers and paste your primer so it's working your primer so here you should put forward primer and add primer and now we need a reverse primer so this is the reverse primer copy this primer then go to the snapgene and open the primer and then add the primer and there paste and you can see here it's working and here you can see a reverse primer so you can put here or and add primer so during the lecture I told you that these red colors are showing you can see we have a vector and now we prepare a gene fragments and now we need to add this red color means restriction site so now we are going to add the restriction site and restriction site we will pick from the vector so now I open the pet28a vector and you can use the restriction side you can see in CS regions and here you can choose these are all the restriction sides you can choose just to suppose we are choosing the NDE1 and you can go to the sequence and this one is the NDE1 so for the forward we are using the NDE1 for the forward primer so in case of farmer you can see this arrow if you are choosing for forward primer you must shoot the top strand sequence and it should be around 15 nucleotide base pair from left side and 15 from left side and 5 from right side so it should be total around the 20 and which you choose the 5 from this side right side you can choose 4 5 6 7 8 10 mean they are doesn't matter because they are digesting they are digestible so I will choose the 4 sequence but we can choose also 4 or 5 so control C and control F then you can see the green portion we have choose and this is around 20 nucleotide sequences here you can see the base pairs and then go to the fragments and right click on the primers and edit primers and you should put your restriction sites on the start of your forward primer and then paste here and you can see your restriction sites are binded so here we should mention the name we add the NDE1 primer, NDE1 restriction sites, and for next one, we can choose any, but if we are using the his tag, so we should choose the any the restriction sites which should be near these regions. So we can choose any one here. So I would like to choose the XHO1. So in case of forward primer, which we will choose from this side, upper arrow. But in case of reverse primer, we will choose the bottom strand and we will choose the 15 amino acid from the right side. In case of forward primer, we are choosing from the left side 15 in this direction. But in case of reverse primer, we are using the right side 15 amino acid, and 15 we can see here, and these red are 15 and 5 from left side, but it's not defined just 5. You can change 4, 6, 3, it's all are okay. And then we will go edit and copy the bottom string and 5 to 3, then control F and control V and press, then we can verify our relevant sequence is selected. And now we will go to the fragment and click on right click on the reverse primers and open it and paste your sequence, restriction sequence. And now here you can put which sequence we have used, XHO1, XHO1. And okay, so now our fragment is ready with restriction sites. And now next step is PCR. So now we will open the actions and then press the PCR, and then here we bind the primer first, forward primer, and then reverse primer. And here we can see it's a green, so it's mean our primer and restriction sites are working very well. And our fragment size is 800 base pair almost. So we will name this the PGM1 and 001. Okay, then we can see the over sequence here. And now this sequence is, now this fragment is ready for insertion in the PET28 vector. So now we will open the PET28 vector and then go to the action, and then gypsum. Here we will use the gypsum method. There are many methods like conventional method, gateway colony method, and gypsum colony method. So we will use the gypsum assembly method. And here we have just one fragment, so we will choose the just insert fragment option. If you have many fragment, then you can choose the second, but we have just one fragment, so we will go to head with one fragment. And here we will go to the linearize with the restriction enzyme, because we are using the restriction enzyme. So our first restriction enzyme was the NDE1, and the second restriction enzyme was the XHO1. And here you can see our gene of interest will be fixed here, and then we need to choose the fragment. And this one is our fragment which we have prepared now. And then we should choose the ND1 primer, and this is the XHO1. And here now we can see our fragment is ready, our construct is ready. And here we can see the all the fragments restriction side. We have choose the 15 and 15. Basically we choose one for 19 and second for 20, but here we can see just 15 15 in a base pair, because we choose the 5 or 4 which I told you from the left or right side. They are digestible portion, it doesn't matter. But which we choose the 15, it's very important. So we choose the 15 from reverse and 15 for forward restriction side. And now you can see our fragment is ready. And now we will label with the specific name. Suppose PGM1 construct and then assemble. So that's our fragment, and here when we go to the sequence, and we can see here our fragments. And one thing is very important. So this is our fragment, our gene of interest, this green color. And when I choose the ND1, you can see here no extra nucleotide amino acid. This G and A are bind with each other. There is no extra amino acid, but mostly beginner students are making the very common mistakes. They are using here another extra amino acids and extra nucleotides that are not good. So here must should be remove the extra amino acids. And similarly we will check the XHO1. You can see XHO1, there is also no extra amino acid, so it's mean our construct is okay. So if you have any question, please ask. Thank you so much for listening.
Thank you so much, Mr. Naeem Khan, for today's insightful session about uh, gene construction. It's very wonderful and very important lecture regarding yeah, regarding molecular biology. So, if you are biotechnologist, it's very important for you. So,
Yeah.
if you don't understand or skip or join late, you can join it. Uh, you can take that lecture uh, whenever you free. Okay. So, if you have any question now, you can write your question. And first and last point is that during the session, don't any irrelevant comment in comment section. Expect your roll number and your name. Okay, so it's very non-serious behavior. This you are during the lecture you are answering question to each other. It's the first and last reminder. Don't do this again. Okay. So if you have any question, you can write your question. Mr. Naeem is here. He will answer your question one by one. Thank you for cooperation. Best regards. Please off. Please off phone. Please. Dear Mr. Naeem, here is
Yeah,
one question. My voice is
yeah,
clear to
yeah,
you.
yeah, you can ask.
Here, how much it cost for primer and how we
Uh,
can get best primer from NCBI primer designing.
okay.
Can I
So first,
repeat?
yeah, no, no, it's okay. So first, your question about the cost. So it depends on the country. In China, it's very fast and it's very convenient because here is just 12 to 15 RMB. We need to pay this type of primers. And in Pakistan, I don't know about the cost. But in China, it's very easy and very quick. Within 24 hours, we can collect our primers. But in case of Pakistan, we need a long time. And the price is also varying according to countries. And it's also different on the nucleotide basis. If your nucleotide bases are just 15 or 20, then you should pay a little amount. If your primer is very long, then you should pay more. That's why I choose 15 amino acid. Even you can choose 20 or more than 20, which I explained during the work at this place. Why we choose the 15 amino nucleotides here? Because if your work 15 is also working in the same way, and 13 amino acid nucleotides also working in the same way. So then if we choose the 30 amino acid 30 nucleotides, then we will pay the double amount, and our work is same. So that's why we will choose the 15 amino acid, and 15 amino acid is the minimum, and in this way we can save the some money. So that's why I choose the 15 amino acids here. And your second question? Hello, hello, are you hearing? Hello, can you hear me? Hello, 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 hello.
Am I audible?
Yeah, now you are available, but I explained you, maybe you are offline.
Yes, some network issues. I am
Oh,
in village.
so, uh, so I should explain again.
Uh, I am, I am, I am explaining next question. Okay.
Oh, okay, okay.
Uh, why we choose primer of small size by nucleotides or less?
Because it's specific. If your size is very long, then it will bind many places. Okay, because you can see here the region is long. If your primer is very long, then there is chance it can bind at many places. But if it's specific, then it can't bind at other places. Specific, it's short mean it's more specific, and short size is around, it should be 18 to 25. But we are using the restrictions colonies, it's more specific. So there is no chance to bind for another. And snapgene have the ability, it will tell you if your primer is binding at multiple places. It will tell you your primer is going to it's going to bind with multiple. Another question.
Okay. E- One is question: is any particular reason for selecting the Gibson assembly method? I mean, how will we
uh,
know when to use which method?
because if you are using, there are three common techniques. Conventional is very old, it's not specific as compared to gypsum is more efficient than conventional. And third is the golden gate is using for the multiple fragment. If your fragment is around 300 to 1000, then you must use this gypsum assembly. So mostly fragments are less than 1000 nucleotide base pair and more than 2 or 300 base pair. So according to the size, this gypsum is more better than the other mm, techniques, like say conventional or second golden gate.
Okay. If anyone have any question more, you can write your question. Or if you want to come on as a host, if you have solid question, then you are also welcome. Okay. and must like any.
Yes, sir. There's a question by a candidate named Fatima. How to choose an appropriate vector?
Yes, it's done,
Sorry.
already done.
How? Okay, sorry.
Uh, here, here question is, how does hairpin structure making?
Hairpin structure is making your primer shouldn't bind with each other. Dimers and hairpins, it's mean. So in Snapgene will tell you if this kind of issue you have. But Snapgene is very efficient tool. If it didn't tell that, so it's mean there is no problem.
Okay, all right.
Yeah,
Thank you
so
so much.
these kinds of basic problems, these efficient tools are telling us.
If your
So there.
primer, yes,
Yes, sir.
Miss, can you kindly read last questions from like Sif?
Okay, so
How many nucleotides?
there is a question by Mr. Malik. He's asking how many nucleotides should be an ideal primer have.
18 to 25.
Okay, there's another candidate, Mr. Mohammed, who is asking, what if our primer is custom made and never used before? Then what annealing temperature will we use?
Annealing temperature mostly Snapgene will give you. You can see here. Here you can see it's giving you temperature. But there are many tools like Amplifix. You can use the Amplifix and check the temperature. You
Sorry,
should.
sir, I guess you were not audible in between. So can you please once repeat?
So I said Snapgene will tell you about the relevant temperature. So it's telling you 77 and 74. So according to my experiments, so I was dividing both. Suppose 77 plus 74. I was uh, add them and so which I collect the sum. Suppose both have the 75 and 75. Then first I calculate the sum of them and then divide by 2, and then I have which answer I have then I was using. But you can use the Amplifix software. It will tell you which annealing temperature you should use. Number one, and number two, when you will design the primer, the company will give you the annealing temperature. And the third one, you can use the relevant paper. Papers are more better than these techniques.
Okay, thank you, sir, for answering it too well. And there's one another question, which is why it is important for forward and reverse primers to have similar TM values.
You can't calculate one by one. It depends on both calculations. Have you used the Amplifix software?
I guess the candidate would have not used.
Yeah.
I would
So
recommend to have the suggestion which is given by sir, and you can always revert back to sir. We can make sure that if you're getting any problem regarding the same. So
uh,
there's another question by Ms. Nasifa. How do you determine the optimal melting temperatures for primers?
So if ingredient temperature, ingredient PCR is better to optimize for optimization. You
Okay,
know
thank you,
about it?
sir.
Yeah.
Yeah, yes, sir. You can proceed.
So I said ingredient temperature is more suitable for everything because there is no other options to optimize. Otherwise, in case of ingredient PCR, you can apply many different temperatures in same time. But if you are going to one by one, it will consume too much time and resources.
Okay, thank you, sir, for explaining it. Miss Fatima, you have a question which has left unaddressed. Kindly, please, you can retype it. Till that time, I have a question from sir. Sir, as per your experience, what uh, difficulties you have faced in softwares you have used for primer making, and what do you think which softwares are the most reliable and most accurate for making primers, since it plays a very important role in any biotechnological amplification, etc. So if you can answer it.
I think Snapgene is better. There are many tools like CLC and Snapgene, but Snapgene is better. I would like to prefer to use and recommend to you. You must should learn the Snapgene.
Thank you, sir, for answering it.
Yeah.
Miss Fatima had a question. Uh, like she asked, how to choose an appropriate vector for amplifying our gene of interest. If it is answered already, I would kindly request you to repeat it. I guess
Uh,
Fatima has not listened it.
okay. So when you are choosing the gene and vector, then vector have a specification. When you will go to the website of our vector or from any search engine, you can search suppose PET28A vector. So first I should know about this. If my fragment size is 800 base pair, so must I should know this vector can adapt this size or no. Some vector are using just 2 or 300 base pair nucleotides, but they are not expressing the large fragments. So first is size is very important, and second are the feature. Every vector have a specific promoter. You should read the different papers, published papers, and you should follow them. Which vector they use for which genes, and third one is the strain. Mean this vector. Suppose we use the PET28 vector. We should know this vector will express in bacteria or yeast. It's also very important. Otherwise, and
Okay.
website also mention all specifications and everything.
Yes, sir, the candidates are telling you thank you for the same thing. If there are any last questions, we can wait for 2 minutes. And
Sure.
any last questions, candidates can type in the chat box.
Okay.
Um, yes, any last questions, any candidate, any feedback for the speaker would be appreciated. Okay, the candidates are telling that it will be useful for them and it was an amazing session. I would really appreciate that you have made the session a perfect balance of the practical thing and along with telling the theoretical part and along with answering each of the questions. I'm sure when they will practice, if they have any question, they will reach out to the organization. And if we are not able to solve their queries, so we will always they can reach out to you through us. And thank you so much, sir, for taking time out of your busy schedule and uh, making us aware of the most important part in biotechnological application of any lab, biotech lab.
Okay, at the
Yes,
end,
yes.
there is some important announcements. Focus on lecture during the lecture, and at the end we will make answer question, discussion, panel discussion. Uh, okay, I hope you understand that point. And second is, if you have attendance issue, so kindly clear it on the spot. Okay, so right now speaker is going. Okay, so after the class we will give the question of your answer. Okay, so last announcement is that uh, it's your responsibility attendance. Okay, and we will give just uh, yes, we will you give the certificate of internship. Just those have 95% attendance according to commitments. Okay, so also report is mandatory. A good report will receive also a recommendation letter from the organization, and those are working and those are learning with passion. We have some amazing opportunities and collaborations for those who want to really passionate about biotechnology and its further domains. Okay, so we have a lot of opportunities for you if you have passion of learning. Okay, so at the end, if anyone have any question, you can ask. Okay, must support our YouTube channel. Also share it with your circle, and also like and comment after the session. Okay, so if anyone have no question, you can leave the meeting. Best regards. Allah Hafiz.
Thank you, sir. The questions are all answered. If there will be any, I'll just go through it and the team will reach out. Thank you so much for giving your time, sir, since time is money nowadays. And thank you for delivering the knowledge to the new budding biotechnologists. Thank you a lot, sir.
Welcome, welcome. No issue. Okay, now it's going to end.
Yes, sir. Any last words by the CEO, please? Mr. Gulam?
Yes, you are audible.
Yes, any last words to end the session for the speaker from your side, sir?
We need to conclude it. Thank you so much, dear Naeem, for joining us in today's session. We are very thankful uh, for your time, for your precious time. Also, thankful Dr. Shehzad, who connect us on that prestigious platform towards learning and serving the community. Thank you so much.
Welcome, no issue. I would like to say also thank you so much for you and your team that you provide me this opportunity to explore myself. I am also very happy to work with you. So I hope in future if you have this type of activity, I would serve for you with happy. So if you have this kind of activity, kindly tell me.
You sure? Inshallah, in future we will design some projects and we will work together. Thank you so
Sure.
much.
Okay. Welcome. Allah Hafiz.
Khuda Hafiz. Allah Nigeban.
Candidates, you can leave the meeting. The session is officially over. Thank you everyone for joining in.
Thank you so much everyone for joining. We want same passion for learning till the end of internship. Okay? Best regards. Allah Hafiz. Allah Nigeban.